In this video, I would like to introduce the concept that cancer is associated at the cellular level with metabolic dysfunction, or more specifically, with the reprogramming of cellular metabolism. Let's compare the metabolic needs of different types of cells. Many healthy differentiated cells have a relatively low rate of cell division. One of their main sources of fuel is glucose. And in these cells, the complete oxidation of glucose into carbon dioxide is made possible by the activation of three catabolic pathways: glycolysis, citric acid cycle, and oxidative phosphorylation. In cancer cells, series of mutations impact multiple signaling and metabolic pathways, leading to metabolic reprogramming. Both genetic mutations and the tumor microenvironment alter the activity of metabolic pathways. The consequences are an increased cellular growth and an accelerated rate of cell division. In fact, a similar metabolic reprogramming is observed in healthy cells characterized by rapid cell division. Some of these cells are found in developing embryos. Other ones are involved in the immune response triggered by infection or involved in wound healing. Cancer cells, like healthy cells, are influenced by internal factors as well as external factors. Many types of cancer are characterized by well-described genetic mutations affecting signaling pathways and driving the tumor phenotype. Here are examples of these mutations commonly associated with different types of tumors. Mutations in genes coding for protein involved in the PI3 kinase, AKT, mTOR signaling pathways. Mutation in genes coding for MYC, KRAS, p53 proteins. These different genes code for proteins that are key drivers of metabolic reprogramming in cancer cells. Furthermore, the tumor microenvironment affects cancer cells. This microenvironment is typically hypoxic relative to healthy tissues. It is due to a poor vascularization of tumors. The concentration of nutrients and the pH in the microenvironment impact the metabolism of cancer cells. Metabolic reprogramming can be defined as a set of interrelated metabolic adaptations in cancer cells. There are bioenergetics adaptations to support the synthesis of enough ATP and enough biomolecules to make rapid cell growth possible. Increased biosynthesis impacts the production of the main types of biomolecules: carbohydrates, proteins, nucleic acids, and lipids. Finally, an hyperactive metabolism leads to oxidative stress and the needs to maintain as much as possible the redox status of cancer cells. In this video, the focus will be on bioenergetics. One of the first observations of abnormal metabolism in cancer cells was done by Otto Warburg and is called the Warburg effect. He observed that tumors have a high rate of glucose uptake and release a large amount of lactate. These observations suggested a very active anaerobic glycolysis. However, unexpectedly, this metabolic activity persisted under aerobic conditions. Originally, it was assumed that cancer cells had dysfunctional mitochondria and were unable to complete cellular respiration. This hypothesis is incorrect, since there are very few cases of cancer characterized by non-functional mitochondria. To understand the Warburg effect, we need to compare the metabolic pathways of glucose in healthy and cancer cells. Healthy cells import pyruvate, the end product of glycolysis, into mitochondria for processing in the citric acid cycle and producing reduced electron transporters such as NADH and FADH2. These reduced molecule drives the electron transport chain and ATP synthesis by oxidative phosphorylation. The outcome is the production of 36 moles of ATP per mole of glucose. Pyruvate is converted into lactate by fermentation only when oxygen is limited. In contrast, cancer cells favor fermentation over oxidative phosphorylation, even in the presence of oxygen. An average cancer cell diverts only 5% of its pyruvate into mitochondria, while the rest is converted into lactate. The result is a reduction of ATP yield with only 4 moles of ATP produced per mole of glucose. This observation was very puzzling to cancer biologists. Why would cancer cells give up on the production of valuable ATP? Scientists suggested several answers to this question. One hypothesis was that aerobic glycolysis was a disadvantage for cancer cells, but was driven by genetic mutations that accumulate in cancer cells. Another hypothesis was that the hypoxic tumor microenvironment drove cancer cells to rely on glycolysis. However, other scientists understand the Warburg effect from a different perspective. What if ATP production is not the top priority? Let's look at the production of biomass. In healthy cells, oxidation reactions during glycolysis and the citric acid cycle converts a 6-carbon glucose into 6 molecules of carbon dioxide. Carbon dioxide is released in the blood and eventually exhaled, so the carbon is lost. Now, what does happen in cancer cells? Because a small fraction of pyruvate is completely oxidized, less carbon is lost. The intermediate compounds are used as precursors in anabolic pathways. The retained carbons are used by cancer cells for growth. Lactate, produced by cancer cells, can be recycled in the liver via the Cori cycle and produce more glucose that can be used by cancer cells. Which is more important to a cancer cell, ATP production or biomass? Let's look at the numbers. Cancer cells need to make proteins, nucleic acid, and lipids. Let's use palmitate, a fatty acid found in membrane lipids, as an example. The biosynthesis of a palmitate molecule requires 7 ATP, 14 carbons, and 14 electrons given by NADPH. Let's assume that the biomass and ATP have to come just from glucose. While glycolysis, the citric acid cycle, and oxidative phosphorylation provides a lot of ATP, the carbons from glucose are lost and NADPH is not formed. In contrast, glycolysis followed by fermentation retains carbon and generates a small amount of ATP and NADH. Furthermore, as you recall from a previous video, glucose 6-phosphate, an intermediate of glycolysis, is a precursor of NADPH synthesis via the pentose phosphate pathway. So NADPH synthesis is stimulated in cancer cells. The Warburg effect observed in cancer cells does not sacrifice ATP production. It is about conserving the precious carbon biomass needed for cell growth and division. Let's take a look at the regulation of glycolysis. As you recall, the conversion of phosphoenol pyruvate into pyruvate is one of the 3 metabolic valves in glycolysis. The enzyme controlling this valve is called pyruvate kinase. However, there is not just one pyruvate kinase. There are 2 genes coding for at least 4 forms of pyruvate kinase. These variants are also called isoforms. The gene L is transcribed, and the transcripts are alternatively spliced to produce 2 isoforms. The L isoform, that contains exon 2 but not exon 1, is expressed in the liver. The R isoform, that contains exon 1 but not exon 2, has an expression restricted to red blood cells. Similarly, the M gene produces 2 variants by alternative splicing. M1, the most common form of the enzyme, is found in a large variety of tissues. M2 is a variant that is produced in rapidly dividing embryonic cells and in tumor cells. So how does pyruvate kinase M2 contribute to the metabolic reprogramming of cancer cells? In fact, in cancer cells, the pyruvate kinase M2 is mostly present in an inactive form within the cytoplasm. The consequences are the following: an accumulation of phosphoenol pyruvate that is not converted into pyruvate, an overall increase of all the glycolytic intermediates, and a rerouting of these intermediates to anabolic pathways contributing to biomass production. For example, the excess of glucose 6-phosphate feeds the pentose phosphate pathway, leading to the synthesis of NADPH, the electron donor that is essential for many biosynthetic pathways. The low activity of the pyruvate kinase M2 apparently counters the Warburg effect, with cancer cells producing less ATP and pyruvate. However, the combination between the Warburg effect and a less active pyruvate kinase M2 supports the carbon retention that enables rapid cancer cell growth. So what are the other advantages conferred to a cancer cell by the Warburg effect? The following hypotheses are not mutually exclusive. The Warburg effect provides enough ATP for cancer cell growth in the hypoxic environment existing in poorly vascularized region of a tumor. The Warburg effect triggers a strong glucose and glutamine intake that could starve healthy neighboring cells and thus favor the growth of cancer cells. The increased secretion of lactate by cancer cells reduces the pH of the tumor's microenvironment and limits the impact immune cells could have on the tumor. Finally, the accumulation of glucose 6-phosphate supports NADPH production that is not only needed by anabolic pathways, but is also needed to reduce the effect of high reactive oxygen species in cancer cells. This high production of reactive oxygen species is a direct result of the high metabolic activity existing in cancer cells. It is very informative to compare signaling pathways that regulate the metabolism of healthy and cancerous cells. In healthy cells, glycolysis is negatively regulated by p53 signaling. Furthermore, the LKB1 AMPK pathway suppresses mTOR signaling to ultimately depress the rate of glycolysis. In conjunction, glycolytic precursors are channeled all the way through the citric acid cycle to maximize ATP production. Meanwhile, in cancer cells, many of these signaling pathways are altered. For example, many cancer cells contain a mutation in the gene coding for p53. One of the consequences of p53 inactivation is the loss of a negative regulator of glycolysis. Other mutations found commonly in cancer cells results in the inactivation of the LKB1 AMPK pathway and the activation of mTOR signaling. This activation has several consequences: a direct activation of glycolysis, an increased expression of the transcription factor HIF1. HIF1 expression is also triggered by hypoxic conditions. HIF1 triggers an activation of glycolysis and a simultaneous inhibition of the citric acid cycle that contributes to an increased concentration of glycolytic intermediates and promotes the conversion of pyruvate to lactate. As you recall, the predominance of the inactive form of the pyruvate kinase M2 in cancer cell reduces the synthesis of pyruvate and further contributes to the accumulation of glycolytic intermediates, thus promoting cell growth. Distinct to the metabolic reprogramming that we observe in cancer cells, scientists are interested in cancer cell metabolites, also called oncometabolites. These molecules are observed at much higher concentration in cancer patients than healthy individuals. We'll take the example of an oncometabolite called 2-hydroxyglutarate. To understand how 2-hydroxyglycerate is produced, we need first to recall one of the citric acid cycle steps, the conversion of isocitrate into alpha-ketoglycerate. This reaction is catalyzed by an enzyme called the isocitrate dehydrogenase, or IDH. As observed in the case of pyruvate kinase, there are several variants of IDH, IDH1, 2, and 3. This table outlines the differences between the three variants. While the three variants catalyze the same reaction, they use different cofactors and have a different intracellular distribution. IDH3 is found in the mitochondrial matrix and uses NAD+ as a cofactor. In contrast, IDH1 and 2 both use NADP as a cofactor, but IDH2 is mitochondrial, while IDH1 is cytoplasmic. Mutations in the genes coding for IDH1 and 2 are frequently observed in different types of cancer, including gliomas. The mutant enzymes accept alpha-ketoglycerate as a substrate and reduce it to 2-hydroxyglycerate, a reaction coupled with the oxidation of NADPH. Therefore, the elevated level of oncometabolite 2-hydroxyglycerate in some cancer cells is a direct consequence of the expression of mutant forms of IDH1 and 2. How does mutant IDH activity impact the cell's metabolism? Importantly, the mutations in IDH1 and 2 affect only one copy of the genes. So these cancer cells express wild-type IDH, produce alpha-ketoglycerate needed for the survival of cancer cells, and generate NADPH. In addition, the mutant forms of IDH produced in cancer cells will lead to the consumption of alpha-ketoglycerate, the consumption of some NADPH, leading to an alteration of the redox balance in the cancer cells. The production of an abnormal level of 2-hydroxyglycerate is also observed, and this elevated level makes 2-hydroxyglycerate a very good cancer biomarker. There are some reports indicating that 2-hydroxyglycerate could directly impact the metabolism of cancer cells. In conclusion, in this video, I have discussed the importance of metabolic reprogramming in cancer cells and showed how genetic factors, especially mutations affecting signaling pathways, impact the metabolism.